Hi, everyone. Thank you for joining today. We will get started on the hour. Hello, everyone. Thank you for joining today. Welcome to the NOAA Omics seminar series. I'm your host, Nicole Miller, and I am a NOAA Omics portfolio specialist, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency, collaboration, and highlight the incredible omics-related research currently underway both within and outside of NOAA. The seminars from this series are made available on YouTube and our NOAA Omics website. There's a link to the website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation. And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Exploring Ocean Twilight Zone Biodiversity with eDNA by Dr. Annette Govindarajan. Annette, I'm going to go ahead and share presentation with you, make you a presenter for you to set up your slides. Dr.
Hi.
Awesome. There we go.
Is the slide showing? Excellent.
Yes.
Thank you.
Dr. Govindarajan is a molecular ecologist in the biology department at Woods Hole Oceanographic Institution. Her research focuses on understanding the biodiversity, ecology, and evolution of marine animals, especially in the midwater realm. Towards these ends, she develops and applies environmental DNA approaches, including sampling technology and strategies geared towards the deep ocean. Dr. Govindarajan has a BS in ecology from the University of Connecticut, a master's in oceanography from the University of Connecticut, and a PhD in biological oceanography from the MIT Hui Joint Program, and completed her postdoc at Hui. Thank you, Dr. Annette, for presenting today, and I will now pass it off to you.
Thanks, Nicole. Thanks for the introduction, and I'm excited to be here to talk to you today about my research on Ocean Twilight Zone biodiversity, specifically diversity of animals using eDNA. So I want to, before I get started, I want to give a big thank you to everybody who's made this work possible. And the work I'm going to talk about is encompassed the last maybe 5 to 6 years. So I have a lot of people to thank from my laboratory and then my collaborators and co-PIs. And this work was funded through Hui's Ocean Twilight Zone program as part of the Audacious Project, a collaborative endeavor housed at TED, as well as NOAA's Ocean Exploration Cooperative Institute. So, the focus of this work is on the Twilight Zone, or otherwise called the mesopelagic. That's the region of the ocean between 200 and 1,000 meters depth, where there is low and decreasing levels of sunlight. A lot of many of the animal species undertake diel vertical migrations, where the typical pattern is that they would come up into shallower waters at night to feed and then head back down to deeper waters where they would spend the day. These animals, through their feeding and their migratory habits, can have a big impact on the carbon cycle through the biological carbon pump. However, there's a lot of unknowns about these animals, and a lot of that has to do with the challenges for studying this environment. And so I have this quote here from a paper that was influential at in the beginnings of our putting together our OTZ program from St. John et al. There is a major lack of knowledge on the global composition and distribution of mesopelagic diversity, which is undersampled and sparse in data. So, so that's sort of the framework that sets up our overarching science questions, which include what are the species that comprise the OTZ, and we say OTZ to mean Ocean Twilight Zone. It's just a shorthand for that mesopelagic region. What are the species that comprise the OTZ biomass, and how are they distributed in space and time, and the factors driving those distributions, in particular, what species are undergoing diel vertical migration, and what are the patterns and timings of their migrations, and how can we observe and monitor biodiversity changes that are related to climate change and ocean health. So these are some big questions that, there we go, that we want to answer. And to do that using DNA-based approaches. And so we have what we call barcoding technology, where a barcode is a short stretch of DNA that in a particular marker that for individuals within a species is similar or identical, but different between species. And we can do bulk sequencing so that we don't, not just for individuals, but for entire communities. So you can take the contents of a plankton tow, for example, and grind those up and get hundreds of thousands of sequences that represent everything that was in that sample. And then with eDNA, which stands for environmental DNA, we can actually just, we don't need the animals themselves anymore. We can just sample for the trace genetic signatures that they leave behind as they move through the water. And so for animals, this can be, sloughed off cells or fecal pellets or gametes, or come from partial predation, or so a lot of different mechanisms can result in the production of these traces of DNA. And so we can access that by filtering the water and collecting the DNA from the filters. So rather than the animals themselves. So this slide shows the general workflow for eDNA. First, we've got to get the samples. And so the typical way that's done for Ocean Twilight Zone and deep sea research is you would go to sea and you would collect the water with Niskin bottles. Usually those would be mounted on a CTD rosette. And then you bring those water up to the ship, and then you filter the water onto little filters, which you would then preserve. I'm going to also talk today about our development of autonomous samplers that can do the filtration in situ, and that can be mounted on different types of exploration platforms, like what's shown here is the robot Mesobot, but these samplers can be mounted on different types of platforms. So we collect our samples, we take them back to the lab where we do our DNA extractions and sample processing, and we then sequence the barcodes. We call this is the meta barcoding, and we get back our hundreds of thousands of sequences per samples. And then what we have to do is assign called taxonomic assignment by comparing our data to reference barcode libraries that contain sequences from specimens that have already been identified, that we're confident in the identification. So that's the workflow. Now, eDNA for animals is still a relatively new approach in the ocean, and particularly so for the deep sea. And so this is a figure here from a recent review by Patton and Goodwin that shows Google Scholar citations for marine environmental DNA. And I pointed out that the year of the start of our OTZ project, but and you can see that those are increasing, the publications are increasing rapidly. But if you actually try to narrow that down and say, well, what about, for the mesopelagic, and then what if you narrow it down to animals? Because actually a lot of this kind of eDNA work is microbial focused, right? Then it really narrows down quite a bit. And then in fact, if you actually look at try to find papers that actually were experimental mesopelagic studies using eDNA, in 2018, there's really none. So none that I could find. So it's really a new approach. So towards that end, when you have a new tool for approach for studying the environment, you have to think about how to interpret that, right? So what we've done in my lab is developed a framework where we want to look at our eDNA results and understand how to interpret that, just like any sensor, really, right? It could be you get your results, right, and then you compare that to the actual ecosystem, and they're different. So if you look at the top part of this figure, on the right-hand side, we get the ecosystem inferred from eDNA results, and you see that there's different relative abundances of represented by the different colors, right? And then if you were to compare that to what's actually out there, you see that doesn't agree, that there are some differences, right? And that it's not exact. So what we want to do is understand how is what we're observing differ from the actual ecosystem. And so we can think of that the kinds of factors that bias our interpretation. We have environmental factors, Way to think of these is the ecology of eDNA as well. Like how do animals shed DNA? How does that DNA get transported and dispersed through the environment, and how does it decay? And so all of these factors might be different for different types of animals, and so all of these will impact the DNA signal in the environment. We also have sampling factors, how much sample that you take, how large of a volume, how many samples, the way the sampling strategy was designed. These things can impact how you interpret the data. And then we also have laboratory factors. And so things like your protocol choices in the lab can have a big difference on your results. We also need to have that reference barcode library for taxonomic assignment, and our informatics steps can also lead to the way we view our data. So basically, in our lab, we've looked at many of these different environmental, especially environmental and sampling factors, and some of particularly the reference barcoding to try to understand how we can have this framework for OTZ analyses. So first, when we first did this work, the first cruise we had was in 2018 on the Bigelow, when we went to the North Atlantic slope water. We took some CTD casts where we took the water and sampled it. And then we also did a net tow using a mock nest, which can give you vertically stratified net tows. It stands for multiple opening closing environmental sampling system. And so we compared the metabarcoding results using a genetic marker that would reveal a wide range of eukaryotic diversity. And we compared our eDNA results with our net tow results. Okay, and again, the focus here was, my focus is on animals, and so that's the part that I looked at. And we see, just by looking at the colors, you can see that they're quite different. So the eDNA has a lot more of the gray, which are the siphonophores, so they're gelatinous species, and then the mock nest has a lot more of the yellow, which are the krill or euphausids. So those are crustaceans, and so they're different. And so on the other hand, we're also comparing apples to oranges because they're two different entities, right? DNA signatures versus actual organisms. One thing we didn't see with this particular marker was fish, and so I'm going to circle back to that, and that's an example of how your primer choices can influence what you see. But we'll come back to that in a few minutes. One of the things that we saw in this early study was that when we think about the number of taxa that were detected, right? Even though the mock nests had greater number, but if you think about it in terms of sampling effort, the eDNA was very efficient, right? Because the eDNA water volumes were so small compared to what the nets were sampling. So what was striking to me was how efficient the eDNA could be at detecting animals. We also saw that there was some significant differences with depth in terms of the depth categories that we saw, especially between the surface 0 to 100 and deeper. But even in the deeper water, we did not see differences in daytime and nighttime casts in this particular study, but again, that's something we come back to, because that has to relate to, I think, how the study was designed and what we were looking for. Okay. One of the key questions with eDNA analyses, though, is we can get these signals, and we're detecting a lot of animal species, right? But how do we know, how can we be sure that the eDNA signal that we're detecting comes from where we're sampled, our sampling location, or could it come from somewhere upstream or up in surface waters, right? And so how does eDNA move through the environment, and how does that reflected when we detect it? And so this work was done by a postdoc in my lab, Eileen Allen. She did a modeling study, also with our co-authors, and she specifically looked at the role of vertical migration in looking at the vertical distribution of eDNA in the vertical dimension. And the good news was that the eDNA signal appears to be localized within 20 meters or so of where it was shed. And so that's telling us that eDNA can be informative about patterns and processes in the vertical dimension. This paper did not look at the horizontal component though, but for the vertical, where we're very interested in processes on the vertical axis, that was telling us we can, detect processes like diel vertical migration. So that was really exciting and validating for the approach. Coming back to the detection efficiency for eDNA, as I mentioned, it was highly efficient given that the volumes were small, but maybe we could need to get larger sample volumes. Okay. We also found, you have a very limited number of Niskin bottles on a rosette, right? And you might want to take more samples. So that's a limiting factor as well in terms of how we do the experimental designs. The process of filtering the water is labor intensive, and this comes from this picture here comes from a cruise where we actually had like 11 pumps set up and being filtered at the same time. We call this our spaghetti slide because of all the tubing looked like spaghetti. And so you can imagine, it could easy to get something confused, make a mistake. And there's also, the more handling, the more potential pathways for contamination. So these are some limitations that led us to motivate us to develop autonomous sampling approaches with in situ filtration. And so this work comes published last year, shows our first version of a eDNA large volume multi-sampler that we put on the midwater robot Mesobot, and did this work in collaboration with Santiago Herrera, Jill McDermott, and Dana Yerger. And we did these deployments in the Gulf of Mexico, and we compared the large volume sampling with the typical CTD sampling approach with the Niskin bottles. And so our hypothesis was that we would detect more taxa with large volume sampling than with the small volume CTD sampling, and that the taxa in the small volume CTD samples would be a subset of what we found with the Mesobot large volume samples, and that if community differences though were driven by the more common species, we thought that perhaps that the community structure could be detected using both approaches. So those were our hypotheses. We conducted the sampling, as I mentioned, in the Gulf of Mexico. We had 2 sites, Bright Bank and what we call a slope site, and we took with the large volume sampler, we took 40 to 60 liters per sample, and so the Niskin bottles were only 2 liters, and so so those were our volume differences. One thing that we found with both sampling types was that there was this relationship between the eDNA concentration and depth, and that the eDNA becomes very dilute once you get into the deeper waters. And this was something that we had noticed in our Bigelow data set as well, that the signal becomes very dilute. And so this is, again, further evidence that we may need to do the larger volume sampling in the deep ocean. What we found was that, in fact, we did find more biodiversity using it as represented by ASVs in the large volume Mesobot samples, which are represented by the triangles, and then compared to the small volume 2-liter CTD sample. So this was true in both of our sites, and supported our hypothesis that you sample more, that you're going to find more. We also found, again, that the CTD was finding, those samples were really finding taxa that were a subset of what the large volume Mesobot samples were finding. So again, that hypothesis was supported as well. There were more unique taxa in the Mesobot samples. And then we also are hypothesis 2B, our third hypothesis, that the depth-related community structure could be detected using both methods, and we did find that as well. So anyway, so that work was strong motivation for us to continue with the large volume sampling, and I'll note that this is geared towards animals and not microbes, but so we've expanded on this work now with more updated versions of our multi-sampler and integrating those samplers with diverse oceanographic platforms. So while we started with Mesobot, we have now put them on other vehicles, other like an ROV or towed instruments, and we envision in the future, moving forward, that these will be part of standard equipment on all platforms, right? And so there's a lot of advantages to doing this. You can utilize the platform capabilities and its co-collected sensor data. It's tailored towards the deep sea animal needs, and I think it's more scalable because what we really need, if you think back to that original quote, we really need to get more data and fill in the gaps and get better spatial and temporal resolution in our sampling. And by moving in this direction, that's a way that we can do this. So as we do start integrating onto different platforms, and it's interesting to think about, and important to think about how comparable our approaches are, because there are some differences. When we put the sampler on Mesobot, this was a robot designed specifically to be able to track and follow animals and particles, and it can be Lagrangian, meaning that it can track with the parcel of water. And we use that sampler with a high flow rate, so we can actually stay within a given water parcel. And you can contrast that with other types of sampling that might be Eulerian, where the water flows past it. Okay, snapshot, as this is the Niskin bottles, which essentially snapshot and show just a snapshot. Whereas other types of sampling, like net tows, or if the instrument sampler is towed, it would be integrative over a longer period of time and space. And so anyway, it's important to think about, when you're making comparisons, what exactly your sampling was actually sampling. So we have this diagram that sort of summarizes that, whether something's Eulerian versus Lagrangian, instantaneous like the Niskin bottles, or integrative if it's cutting across scales of space or time. Another important thing to consider with sampling is adaptive approaches, because we know the biomass is not distributed evenly, and we want to focus on the interesting parts, and in particular, acoustics can be, is an important way to determine, the presence of the biomass. And so what we're interested in developing adaptive sampling approaches. We've done this with where we can control the sampler from the ship, from a laptop, in response to real-time observed acoustic backscatter data. So we can say, oh, we're in an interesting layer, and push the button and start taking the sample, right? And so we can take samples above, within and below layers, and use that the goal is to use that to determine the composition of that biomass. We're working on an interesting new approach of adaptive sampling, where we use multiple vehicle coordination, and this is with the uncrewed surface vehicle DRIX and Mesobot, where DRIX, the little red vehicle there you can see in the picture, can send basically texts Mesobot and tell it to where to go. Okay, so we can detect the acoustics with DRIX. That information is conveyed to a team that then tells DRIX to tell Mesobot where to go, and we're working on further developing that. Another piece of the framework that we've been working on is the reference barcode libraries, and we've been focusing on particular on fish, and So it's very important that we have specimens that for our references that are accurately identified. And so we use what we call an integrative taxonomic approach based on multiple lines of evidence from genetics and morphology combined. And so we have a couple works, one recently published, one in review, where we've barcoded altogether 80 species of fish, unique species, collected by my collaborator, Joel Yopes, and we've identified them with morphology. They're georeferenced and with a photographic library that's corresponding. And a subset of those, we've also characterized the otoliths, which are the ear bones. And those also have been sequenced for two different markers, the CO1 and 12S, which are common barcode markers. We've seen some interesting findings with this, is that when we do compare our sequences to others that are out there on public databases, sometimes there's mismatches. So there's clearly, as many of us who do this kind of work already know, that there are some errors out there. And so we got to work to refine that. And it also can lead to awareness of cryptic lineages that maybe when the species look similar, but in fact, at the genetic level, they may be pretty divergent. This work also stresses that even though we're doing this molecular stuff, we still need to have the traditional taxonomies to make sure that our identifications are correct. And anyway, so in one of my first slides, I showed you our first attempt in the Bigelow 2018 cruise, where I used an 18S marker. That we chose that because it amplified a wide variety of taxa, but we didn't find any fish, right? So that was perplexing because we know there's fish down there. And so now we reanalyzed that sample set with a different marker, 12S, which is geared towards fish. And in fact, we found lots of fish. So that would be an example of how a molecular protocol choice can influence what you see. But in any case, we did our comparison with the eDNA and the net toes, and as before, we found that both approaches discovered many of the same species. Each discovered others, some that the other missed. A lot of species that were missed by that eDNA discovered that were missed by the mock nests. We do know that nets actually miss a lot of species that can avoid getting captured. So that's not too surprising. When we look at what species are found in what depth category, we see that for samples that were taken at night in the shallowest depth category, 0 to 100 meters, we find that there's a lot greater diversity than for samples taken in the day. And that's consistent with what we'd expect for diel vertical migration. So I think what we're seeing is that we're being able to detect who these migrating species are. So I have just a couple more slides to show some work that's in progress that we're taking advantage of this framework and some of the technology. We have again the sampler in Mesobot, and what we've done is taking advantage of the fact that it can stay within a parcel of water. Is we've done a time series with it where we've taken samples continuously before, during, and after a migration event, which you can see visualized on this slide here with the acoustics, right? And so those different colors on the horizontal line are indicating our different samples. So what we're hoping to see is that we'll see specific taxa that are associated with the migration and have a better understanding of the timing and the pattern of the migration. And then my last slide here, I want to talk about a cruise that we're just mobilizing for right now. My team actually, the Hui team left early this morning and on their way to Hawaii for this ocean exploration cruise on the Nautilus. And so they're going to use multiple vehicles and technologies to explore the geologist seamount area. And we're going to put our samplers on a new platform, the Deep Autonomous Profiler. That's Chris Roman's instrument, and that one also has Niskin bottles too, so we can do some comparisons there. We're going to take some samples with Mesobot and do some of that DRIX Mesobot adaptive sampling coordination. We call that verified directed sampling. And so the Nautilus, you can see through telepresence, you can see that live streamed. If you're interested, you can sort of follow along the progress of that cruise. We're really excited about it and to see what we can accomplish there. So with that, I'll take some questions. Thank you.
Thank you, Annette, for this very interesting presentation. We do have a few questions in the chat that we'll go over. And if any attendees has any more questions, please feel free to post them in the chat. The first question is by Carol, who asks, in regards to slides 18 or 20, does research by microbial scientists also find less microbial DNA signal with depth? And Annette, I'm not sure that's going to be tailored to your work, but I'd love to get your perspectives on it.
Yeah, so so that's a bit outside my scope. My focus is on the animals. When we've done some looking at the overall eDNA concentrations that we get do decline with depth, and so those would be including all components, whether it's microbial or animal. On the other hand, when we look at the microbial component, which I've filtered out on the slides that I've shown you, but we see more consistency with what we sample, even with the small volumes, than we do with the animals, which are more variable. So that's an interesting difference. And I think it has to do with the fact with microbes, when you're sampling the actual organism, right? Whereas for animals, you're sampling little fragments and pieces and decaying particles that may vary on a different scale, as well as the biology of the organisms themselves may vary on a different scale. So I think there's some differences.
Thanks, Annette.
But I still do think that maybe there is a decline in signal with depth overall. So, yeah.
Thanks for that perspective. We have a question from Sandy, who asks, will we, or how soon will we be able to identify new animal species by eDNA?
So I think you can't identify, you can have a sequence that you don't have, that you can't put a name on, right? And if it's a new species, that's what it would show up. If it's something that's undescribed to science, right, you're gonna not be able to find a match for it. So you'd still need the specimen, right? You can find sequences that you can't match. Those could be just species that are already described, but there's no reference sequences for, or they could be new species that no one's described yet. But it is important to remember we still do need the specimens.
Thank you, Annette. We have a question next from Kelly, who asks, can this framework be used for monitoring the offshore wind turbine monitoring work being done on the East Coast, thinking about pre versus post construction community changes?
Yes, I actually think that eDNA is a great tool for that challenge and for observing in general, because I think it's a way we can get efficiently get a lot of data, and I think that's a great application for it. I know there's a lot of interest in people doing that, and yeah, absolutely.
Thank you for that. We have a question from Jeff, who asks, can you describe progress towards reliable biomass estimates from eDNA metabarcoding?
Yeah, that's a harder question. And so making to whether you can interpret the metabarcoding in a quantitative way and relate that to abundance or biomass. And I know that that's an active area that people are working on. and would like to do. So it's, maybe there's some progress there. It's not what I'm specifically doing though.
Thanks. Thank you for that. Next, we have a question from Mark who asks, how do you filter very large volumes without clogging? Is there ever too much organic material to process with typical extraction? A related question is, what is the water volume equivalent typical PCR reaction? We'll start with the first one.
Yeah.
How do you filter very large volumes without clogging? Is there ever
So
too much organic
they
material?
generally don't clog. That's the thing. I think that's speaking to the fact that it's dilute, right? We really haven't had clogging be an issue. We do use large area filters, so there's a bit more area to collect material on, but we haven't had an issue with that, right? So I think that's interesting and speaks to the need for that it's dilute down there, and that we do want to get the material. I think speaking to, I think that the latter part to the questions about getting the PCR and everything. So what we do is we'll take the filters back to the lab, and then we extract the DNA. And so we've done a little bit of work where, so what we do is we can cut up that filter into different portions, and what we've been doing is extracting all of the portions, and we've done a little bit of comparing these different filter slices, and because if one slice could be representative of the whole filter, and it actually looks like for the animal component that there's some variability there. So then so that's kind of leading us to want to extract the whole filter, which uses a lot of resources and time. So you can think about maybe some other approaches, like using pre-filters to maybe screen things out so that your inner filter might be more homogeneous, but then what's going to happen is that you're going to collect different types of organisms. There's going to be different types of signals on the outers and the inner filters in that approach. So yeah, it's something that we think about a lot. But what our approach has been right now is to extract from the whole filter, even though it's a bit of extra work.
Awesome. Thank you for that perspectives. Lots of questions coming in. Our next one is, in your fish data, can you tell how much of the mismatch between the plankton nets and the eDNA was adult fish?
Adult fish? No. So that's something that eDNA cannot tell you, right? It can't tell you the life stage of the organism, right? So if you have a larval stage versus an adult, or a juvenile, that you can't tell. So
that brings me to a question I have, Annette, is it possible when sampling for eDNA to differentiate the age of that, or the time length that eDNA, since it was sloughed off in the water column?
Yeah, so that's another interesting question. So you can look at maybe the how big the piece of DNA is, whether it's degraded into smaller pieces. Another approach that people use is eRNA, right, which would reflect what was alive at the time of capture, right, at the time of sampling, because that degrades quickly. So that would be an interesting approach to compare the two.
Definitely. Looks like we have 2 more questions today. So one more from Laura, who asks, how do underwater factors like the Gulf Stream affect results? And how easy is it to map your results geographically?
Yes, that's a good question too. And so I have some data from, it's actually not eDNA per se, it's from the Mock Ness, where I'm working up from a cruise we did in 2019 where we had a number of tows in 2 different sites, one of which was influenced by a warm core eddy and one was outside that sphere of influence. So you could see that even though they were relatively closely spaced, that the water properties, were temperature and salinity were different between the 2 sites. And so you can see that there's differences in overall diversity and what I'm in the process of analyzing is looking to see if those depth distributions are shifted with the between the colder and the warmer water masses. So I think that that's a really interesting question, and hopefully, we'll get some insights on that with our data.
And another question I'm out of my own curiosity, Annette, is where are you most interested in sampling these types of biodiversity estimates geographically in the world?
Geographically, honestly, I'm really interested in almost everything, right? I have to say, I'm very focused right now on the question of diel vertical migration because it's very important process. It's, referred to as the largest migration on the planet. It's really cool to think that these animals can undertake this. It's important to understanding the biological carbon pump, so there's implications there. So that's really exciting. I'm also interested, in understanding how, oceanographic features like the Gulf Stream and eddies can influence distributions. I'm interested in exploring new areas that aren't well explored, like where we're going with the geologist seamount. So I guess my problem is, I have pretty wide interests, and I feel like there's so much to learn still that's okay, that we're going to find interesting stuff wherever we study.
Yeah, thanks for that perspective. Yeah. Okay, let's moving down the question list. This one's from Kim. He says, thank you, Annette. You mentioned the increase in species detected with increased volume filtered. Is this reflected in a very long tail of rare species? Do you think the increase in total DNA captured has any negative effect on amplification, i.e. increased inhibition or template competition?
Yeah, so I think that the increased detection was due to the rare or less common taxa being detected, and whether having differences in template diversity impacted that particular study, I'd say I don't think that it did in my, because we didn't have that issue, right? So I'd say, that wasn't the case, but I have seen, I will mention a little bit technical with a different, when we use the CO1 marker, I have sometimes some inconsistent results where, I think that template diversity is an issue, right? So I think there's some specific marker-specific issues, and that's where protocol testing is important.
Mm-hmm. Most definitely. This next question is from Diane, who asks, can eDNA tell the sex ratio of fish or other animals sampled?
No, so the eDNA, the markers that we're using would be found in all of the population. So we wouldn't, you can't get that information.
Not yet. Hopefully in the future. Our next question is from Allison, who asks, have you tried freezing the sampled water for later filtering? Does this degrade signal?
We have not tried that. It's actually easier to filter on board the ship, because then it's easier when the cruise is done, to take home the little filters as opposed to larger volumes of water to take that home with us. So I think it's it's just what we typically do is when we filter on the ship, we put our filters in the freezer, and then they can all, usually fit in 1 or 2 boxes, which we can ship on dry ice, and that would be just logistically simpler than shipping frozen water. So that's what we do.
Alrighty, we'll take 2 more questions. So our last one is from Daryl, who asks, is eDNA being used to understand and inform potential impacts to deep sea habitats where deep sea mining is being proposed? And I know we touched on earlier just slightly with understanding offshore wind turbines and how we can use eDNA to assess biodiversity there. But what other deep sea habitats where might we be able to use eDNA to inform potential impacts?
Yeah, so I think deep sea mining is a good approach. You can use eDNA to look at impacts of aquaculture. That's something, elsewhere where we might want to look at the impact of biodiversity. We might want to look for, protecting important habitats like deep sea corals, right? So I think all of those are good applications for eDNA observing.
And our last question today, I know you definitely touched on this a little bit, but there's 2. So, is the volume of eDNA collected on a species directly related to their abundance? Or, and another question is, do some species or bigger species shed more DNA than others?
Yes, so this is a good question. We did some experiments on this. Again, that was with my postdoc, Eiley Allen, who did the experiments, and because we were interested in how shedding and decay rates might differ with different animal forms, because a lot of the work tends to be done on fish, but we were interested in what about other species like jellyfish, right, or crustaceans. And so there definitely are differences in the shedding rates and maybe the way that the DNA is being shed.
Awesome. Well, Dr. Govindarajan, thank you so much for your time today. This was a great presentation with lots of interest. We really appreciate it. And thank you all for joining us today. Our next seminar series will be October 18th at 12 p.m. Eastern Time. So please keep an eye out in your emails to learn more about our next seminar series. With that, thank you, Dr. Annette, and thank you everybody for joining today.
Thanks very much. Appreciate being here.